So I was going to focus a little bit on innovation and on the people behind many of the ways we do things. You've had a great introduction here from Sufyan on one particular way that we try to encourage innovation. I should mention I have a conflict of interest list online here at this website in the lower right. I'm going to give 4 quick examples. The first one is on how we can create an organism that has 0 viruses. It has no, as far as we can tell, no possibility of being infected by naturally occurring viruses, even those that we've never seen before. And this has been a saga that's gone on in these papers starting in 2009, where we've done 1, 3, and 7 codons out of the 64 triplets, so all possible A's, C's, G's, and U's coding for 20 amino acids, eliminating 1, eliminating 3, and eliminating 7. And this gives us 3 new functions, which is genetic and metabolic, 4 functions: genetic and metabolic isolation, non-standard amino acids, for example, in this startup company, Grow Biosciences from Dan Mendel. And then finally, this 0 viruses. And I'll just have just one slide showing the 0 viruses after many different attempts. The first one where we got several, maybe 5 out of 7 in 2013 to 16. We now have a way of doing, we think that's general, that should work in almost any organism with the same strategy, which is changing the genetic code of 2 or 3 codons. The key thing is changing 2 codons from serine to leucine. Serine and leucine are very different amino acids, and so when you have the wrong one incorporated, the virus, at every place that it expected to have a serine, it now has a leucine or a mixture of serine and leucine. And so this is fatal in so many ways that the virus can't evolve around it, as far as we can tell. And we confirm this by getting random viruses from the environment, from sewage and from farms, and testing those. And you can see they're undetectable in this assay. And this is Akos, Savina, and Reagan, and others that have contributed to that. Now, that's an example of something that's benefited from the exponentials that we and others have helped bring about in reading and writing DNA, and anything you can make from DNA, like virus-resistant cells. And these are factors of 10 on the y-axis over time on the x-axis, and both reading and writing have improved about 20 million-fold, both in mainly in cost, but also some similar quality improvements, mostly due to multiplexing, not parallelization or automation, but multiplexing, where you mix together samples with barcodes. So, for example, the human genome, which we've helped contribute to the technology for that and interpretation of it, has dropped from $3 billion for an unacceptably poor haploid genome to a modern clinically useful genome around $300. Well, that's great for reading and writing DNA, but what about therapies? Those seem to be going up in price, and some of them are as expensive as $2 or $3 million per dose, for example, gene therapies. And since I'm fairly committed to gene therapies, I felt that we needed to contribute to bringing that price down as well. Before we could even get very far on that, there was a dramatic validation of this premise that you could bring down the price, in that all five of the top 5 COVID-19 vaccines were formulated as gene therapies. They were essentially indistinguishable in molecular mechanism. 3 of them were adenovirus wrapping a double-strand DNA for the spike protein, and 2 of them were similar things with single-stranded RNA. So, but in each case, single gene delivered via a viral or non-viral mechanism. And now the lowest price is $2.2 per dose, rather than millions of dollars. It shows the economy of scale. This is dependent upon having billions of people that were at risk for a pandemic. There are also billions of people at risk for age-related diseases. In fact, most of us, 90% of us, will probably die of age-related diseases. And so we used Pedro de Magales' database. He was a postdoc in my lab, and Noah Davidson searched through 45 candidate gene therapies in mice and found 3 that were worthy of testing further in in dogs and humans. And our tests were not just biomarkers, but first 4 and now growing number of age-related diseases. So if it can reverse the consequences of multiple age-related diseases, not necessarily all of them, but as many as possible, then we're probably getting at the core of aging. And we used 2 combinations of 3 genes in gene therapy. One category were soluble, so they were essentially blood-borne, and the other category were transcription factors. And we'll tell you more about transcription factors in a moment. Anyway, Noah was on these 4 papers and led the founding of Rejuvenate Bio, which is aimed at dogs and human. Now that's large market gene therapies. They've been de-risked on vaccines, but they should also work on age-related diseases. Now, how do we analyze these epigenetic effects that we're harnessing with gene therapies? And the ultimate way of analyzing these, of observing them, is essentially equipping our microscopes so that each molecule we can, instead of looking at it and see black and white or limited number of colors, every protein, DNA, and RNA has its own barcode tag, so it essentially has its own color, and so we can, or it's a combination of colors, and these produce rich 3-dimensional images that can be very high precision, as in the nanometer range. And there's 2 commercial instruments. I think they're quite advanced and mature. One is from the 10x Genomics called the Xenium, and one from Bruker called the Vutara. And our lab and Ting Wu's lab right down the hallway at Harvard have worked closely with these 2 companies. And so I'll just show you DNA, RNA, and protein very quickly. And this is DNA, where you see the 2 pieces of the same region of the chromosome from the mother and the father inherited. These are actually in my cells. So this is DNA from my mother and my father. And you can see, you can rotate them and look at them like any other macromolecule, except these are absolutely gigantic macromolecules that are, if stretched out, would be 2 meters long. But we can look at them with very high resolution, on the order of a sort of nucleosome resolution. So that's DNA. This is RNA, where each dot changes color but not position. Is a single RNA molecule, is a single cell, and then you can have the computer show you that series of washes where you can get a barcode for each of those RNAs. So you know which RNA that is, which RNA splicing or editing or so forth. And this is Evan and Jay did this. And then finally, DNA, RNA, and protein is the last one. Here we can go show the power of super resolution. We can't really tell what's going on with 4 different proteins at once at normal diffraction limited imaging. But if you look at the synapses, you can see those 4 proteins lined up going from presynaptic to postsynaptic. So you know which direction the information is flowing in the brain and at each synapse. And you can tell whether it's excitatory or inhibitory. And this helps you figure out the connectome in a way that was very difficult to do before. Now, finally, I want to talk about machine learning and its application to proteins and then to transcription factors as a particular category of proteins. This is, we not only have machine learning, but we have these mega libraries, which is a kind of quantum computer, is a kind of a biochemical computer where we can make millions of shots on goal. And either the mega libraries of machine learning are valuable in themselves, but together they're incredibly powerful because you don't have to get a perfect prediction in order to get something that's very effective. You're looking for the 1 in a million of the predictions, which is already 1 in a million over random. And that combination is great. And so we published about 8 papers recently. First one was this UniRep, which is a language-based model for machine learning. We did not historically use 3-dimensional structure, even though I'm a crystallographer by training. And then this led to 5 startups. Again, that's kind of the theme here, is innovation getting into commercial, so it can have a positive impact on society. And so NABLA applies this ML to antibodies, Dino to capsid delivery gene therapies, Patch to cis-regulatory RNA and DNA changes. Shape therapeutics is for RNA based therapies, tRNAs, and ADAR, RNA editing. And Manifold is using protein-protein interactions to allow them to barcode possibly thousands of proteins, therapies in a single expensive, say, animal model for preclinical trials, like, say, primate models, very costly. And you could test, in principle, 1,000 proteins at once in one animal. So those are the 5 companies. Here's one of them, just quick illustration. You can design, not mutagenize, not random, but design millions of viruses or proteins and show which tissues they go to, get tissue specificity, get immune evasion. So Pierce and Eric and their colleagues published this and formed Dino Therapeutics. And here's the culmination or an extreme case of that, where you have a key region of 28 amino acids that you want to mutagenize for tissue tropism, say in the capsid. You take those 28 amino acids and our best that we could do in the past, using our very best efforts, we would get close to 0 yield in making a library of 4 mutations out of 28. I mean, that's a lot of simultaneous mutations. If you get to that high a level of random mutagenesis, or even somewhat informed naive models, it breaks the function of the protein of the capsid. But with logistic regression and other deep learning methods, we can get a 90% yield on 25 out of 28, and even substantial libraries with multiple members with 28 out of 28 changes. We've applied the same kind of logic to changing developmental biology, not just proteins that have morphology in the form of viruses, but morphology in terms of tissues and functional differences. We made a comprehensive library of all the human transcription factors and tested them singly and in combinations using transcriptome targeting. We have over 300 recipes, let's call them, where you can cook up your favorite tissue, including some that are now in preclinical trials. And these 4 companies, GC Therapeutics is general, Rejuvenate Bio is aging, or Thymune is for immune tolerance, and Gameto is, as the name says, for gametes. Human gametogenesis. Here's an example of a preclinical trial where we're rescuing a demyelinating disease with a oligodendrocyte-like organoid, which makes perfect wrapping around the axon as you get in the white matter of your brain or the spinal column. Alex and Peristu wrote this Nature Biotech paper and formed a company, GC Therapeutics. So that's all I have to say. Thank you very much, and great success for all your innovations. I'll stop sharing. Thank you.
Thank you very much, George. Very, very interesting presentation. And if here any comments or questions for George?
Or no. Sufyan.
Or Sufyan.
Yeah.
George, if I may, okay.
Well, thank you very much for the marvelous presentation of all these innovations. So I'm actually Mira. I'm the managing director of Nuclear United Kingdom, and I wanted to ask you, all of these marvelous innovations, what would you say are the key factors in the ecosystem that allow the birth of these marvelous companies?
Well, recently it's Nucleate, I can say, but before that it was the Harvard and MIT kind of act as magnets that bring in investors, students, and postdocs. That helps get the thing going, and then the individual lab, like my lab, can get established an own track record over a long period of time, benefiting from standing on the shoulders of the admissions committees for the Harvard and MIT. And then once we get a track record, then we become a magnet or a spotlight for of our own. Also, just an environment where at least part of the goal is thinking about how basic science and technology can lead to something societal benefits. So if you're thinking of all three of those at once, all the time in the lab, you create an environment that's tolerant of small mistakes and failures. And like I said, with our libraries, we have a million failures for every success, and that's fine. That's kind of our metaphor, inspirational metaphor is the library screening. Unlike NASA, where their motto was failure is not an option, we feel that to get really high quality businesses and products, you need to have a lot of failures and a lot of successes. Thank you for that question. It's a good, very good point.
We have one question from our Ministry of Health.
Just a short question. What is the timeline from the idea until the startup gets running or starts running? What is some average time from the initial point until a successful startup on the market?
Well, I'll broaden that question a little bit because you can have a successful startup without having a product. So I would say that having a product is in order. You can even have an IPO without a product. But I think I'll give you a range rather than an average. So the range is probably DNA nanopores, which was a crazy idea in 1987 when we first proposed it, and didn't get patented until 1995. And didn't really have a huge impact as a product until the last few years, where it became the dominant way of getting long reads. Now it has a median read length of 100,000. Put that in perspective, the previous typical read lengths were 300, so 300 to 100,000. So that took basically from 1987 to 2020. A faster one was CRISPR, where the crazy idea was towards the end of 2012, partially inspired by Jennifer Doudna's biochemistry, but requiring a whole different set of mindset to get it into human cells, not out of the in vitro into human cells and then into the clinic. And so from basically January 2013, when we published it, until the start of Editas and Intellia and CRISPR Therapeutics, three companies more or less at once within a few blocks of each other in Cambridge, was just a few months. But then of course, because it required clinical trials, it was quite a bit more than that. The jury is out. I showed you how great gene therapy is, but the editing part of it is still in early stages.
Thank you very much.